slide. I'm acting as the Senior Associate Dean for Research in Global Affairs and also involved in international partnership and innovation in the Department of Internal Medicine at the University of South Florida. I'm also acting as President of the Global Virus Network. And before coming to the direct introduction to Rachel Roper, I just want to emphasize that this is actually a first, which illustrates the partnership between University of South Florida and Global Virus Network. So just in a few words, because some of you may not be aware of this, Global Virus Network is a network now of up to 70 research centers worldwide and 12 affiliates, coordinated from Baltimore in the Institute of Human Virology, headed by Bob Gallo. The Global Virus Network was founded by Bob Gallo in 2011. The missions of the Global Virus Network are really to coordinate research. It's about merging the best experts in virology, working on all human viruses. This is very important. Although today we are going to discuss monkeypox, although obviously we have been very much involved on COVID-19, the Global Virus Network has been and will be working on all human viruses. So we have research, and in particular with task force, again, merging the best experts to design the best activities. And one of the task force is on monkeypox, and Rachel Roper has very kindly accepted to co-chair this task force. We have one on COVID-19, of course, but we also had on Zika, chikungunya, HTLV1, and others. The second mission is education and training with the GVN Academy. You can find all the details on the website, but this is thanks to Liman Lee, who is Vice President of the Global Virus Network, a lot of activities which are ongoing. And finally, advocacy, communication with the different tools. So to close on the Global Virus Network, this is really science-driven, informal network, independent. And these are obviously very important keywords, also very well connected to industrial partners on different programs. So having said that, it's really my pleasure to introduce Dr. Rachel Roper. It's a great pleasure, and we are very grateful to Rachel for accepting to give this lecture. Briefly, Rachel Roper is a professor of microbiology and immunology at East Carolina University. She has been trained in Texas A&M University and then University of Rochester. And then she had a postdoc training in the NIH. And as I said, she's now professor of microbiology and immunology at East Carolina University. Rachel has been working a lot and has a very significant number of excellent papers on several viruses, flavivirus, coronavirus, poxviruses, a lot of work on poxviruses and oncolytic viruses. She's also very much involved in the development of vaccines. She has also been involved in the first sequence and analysis of the SARS-2 coronavirus genome. She serves on many committees, important committees, international committees on taxonomy of viruses, American Society for Microbiology Inclusive Diversity with equity, access, and accountability, and many others. And I mentioned that recently she has accepted to be a co-chair of the Global Virus Network. She's funded by several grants from NIH and NSF. And finally, and not the least, she has been very much involved in gender issues and the positioning of women in science. I will not go into detail, but she has an impressive number of committees she's involved in for this important task. So again, thank you so much, Rachel, for giving this lecture, and we really look forward to your presentation. Thank you.
Thank you very much for that kind introduction. Let me see if I can share my slides here. So thank you, everyone, for attending. Let's start my slides here. So I'm going to talk today about monkeypox virus, the vaccines, and virulence factors for pox viruses. And we have to talk about smallpox, because that's most of the work research has been done. It was really aimed at smallpox, but because they're in the same genus, monkeypox, the drugs and vaccines that work for smallpox are also working for monkeypox. So I'll talk about monkeypox some and the vaccines that are available currently, and then I'll talk about some of the virulence genes that I've studied in pox viruses, both in vaccinia virus, the vaccine strain, monkeypox virus, and these are also contained in smallpox, and how we can use this information to make even better, safer, more effective vaccines. So, most of the research that the world has done on pox viruses has really been targeted at smallpox, because it's such a horrific disease with, 30% mortality rate. Some places they said 50%. It's estimated to have killed about 500 million people in just the first 75 years of the 20th century. Horrible disease. And it was declared a major Category A bioterrorism concern by the US government. And pox viruses are large. Here's a couple of electron micrographs of different pox viruses here. Very large viruses, 20 to 30 nanometers, 200 to 300 nanometers, very complex structure that's very stable. Unlike COVID, SARS coronaviruses that will fall apart over time, the pox viruses are really very stable. And they also have a large DNA double-stranded genome, about 200 kilobase pairs, somewhere around 200 proteins encoded. So it's a large virus with a large genome. And I've been, as I'll talk later, especially interested in what genes might be involved in virulence. So this is the pox virus life cycle. There's a couple different virus types that can enter the virus through a couple different receptors. It really infects almost all cells. And then there's early messenger RNA, DNA replication, intermediate and late messenger RNAs, and then the morphogenesis that occurs in all of this in the cytoplasm, where you get the immature virus and a mature virus, and then it undergoes a double wrapping, an additional membrane wrapping event where it picks up two more membranes, and this is the intracellular envelope virus, which then fuses out and loses a membrane. So this is the basic replication cycle. So smallpox occurred worldwide until the World Health Organization vaccination campaign. It was eradicated in 1979. And this is the famous, first vaccinations, and Edward Jenner showed using a virus he got from cowpox, from cows, that he could protect from smallpox. But there are still a lot of emerging poxviral diseases. Molluscum contagiosum causes about 450,000 doctor visits a year in the US. And we have monkeypox concern, especially now. We had the outbreak here in 2003. Cantagallo, which is actually vaccinia virus, which is the vaccine strain that we use, that circulates in South America and causes outbreaks in humans. Cowpox also causes human infections. And of course, we're concerned about these as bioterrorist concerns. So all of these viruses are in the genus orthopox virus. So they're closely related and they offer cross protection. But with these increasing problems with these viruses, we still need improved vaccines and drug therapies. And there are a large number of unclassified pox viruses that are discovered every year. I'm on the International Committee of Taxonomy of Viruses for pox viruses, a lot of viruses. And we sequenced raccoon pox, the first North American pox virus, and published that. We've done a lot of sequence analysis. So here's just one other pox virus, in the news. So this was a cowpox infection of a human in 2018. And so that made the news in the UK. So again, cowpox is another orthopox virus, very closely related to monkeypox and smallpox. It's usually just a mild disease of cows. It also gets into zoo animals. Zoos see it a lot. And most humans that get it from cats who have been playing with rodents that have it. And so it transmits to the humans. And it's mostly in Europe. So as far as these emerging pox viruses, we have molluscum contagiosum on the right. And this is just, it's not a systemic infection, and it usually is confined to the skin. Doesn't kill people. And then on the left here, of course, we have monkeypox, which we'll be talking more about today. So this came out in 2010 and showed that because we had stopped using smallpox vaccinations, there was an increase in human monkeypox. And so we knew that this was happening. So more and more people were poxvirus naive. So more people were catching monkeypox from wild animals. And we had the outbreak in the United States in 2003 with, I think, at least 100 people infected. And CDC rushed in and was able to clean it up, and it did not become endemic in the United States. So this slide shows monkeypox outbreaks. There were 40 of them in the Central African Republic. So this is just north of DRC, Congo. And you can see there's been a great increase in the number of outbreaks from especially beginning after 2017. The numbers went down here, maybe because of diagnostics, we don't know, or because people were more isolated. But there's been an increasing number of outbreaks, sometimes up to 25 human cases. There were 99 confirmed, 160 suspected, with a case fatality rate of about 7.5%. So, and this is updated as of the end of October now from the World Health Organization, and we have more than 77,000 confirmed cases now of monkeypox worldwide. So it has spread worldwide. And here's a map you can see of cases. The darker areas have more cases. The white areas have no reported cases. Right, but you're only going to report cases if you're looking for them and have diagnostics to detect them. So monkeypox has spread worldwide now in 2022. So these pox viruses, monkeypox has an animal reservoir. Again, it's the same genus as smallpox and vaccinia-virus, the vaccine strain. It can cause a very severe disease similar to smallpox. But it has a lower case fatality rate, about 15% for the clade 1 and about 1% for clade 2, which is the West African, which we're very lucky. That's the one that is circulating worldwide now, the lower virulence strain. So we haven't had that many deaths. And it's mostly in rodents. It's endemic in Africa. And it transmits to humans, especially through hunting, and also rodents around human habitation. So human contact with animals is how it's spreading into the human population. And it's been jumping repeatedly for a long time. So we looked at virulence differences between the clade 1 and clade 2 monkeypox viruses. We sequenced some of these and tried to look for differences to identify what would be the virulence factors. But really, when you're looking at a genome that's 200,000 base pairs and there's a lot of mutations between them, you can't really tell. It's just really hard to do that kind of analysis because it could be a single point mutation. And, with all the changes that are in the genome, it's really hard to track down exactly what is causing the increase in possibly in transmissibility or in virulence. And there can also be changes in the promoter regions that affect the level of protein expression. So we began really seeing monkeypox cases in the news in the US in 2021. And then this one in May of 2022. And this was where we realized that we had a real problem. This person had recently traveled to Canada, not Africa. So we knew it was transmitting in Canada. And then all the cases began to come out, and people realized that this virus was circulating worldwide. And the evidence from the genome suggests it's been circulating since 2017, and we really haven't recognized it until now. So clinical characteristics, vomiting, cough, chills and sweat, headache, myalgia, fever, and what they have here for skin rash is very, very common. And here's the CDC listing for the same thing: skin rash, fever, chills, headache, lymph nodes swollen. And they show, almost everybody has a rash, but I'm concerned there may be a lot of cases that are occurring that we don't know about because the rash isn't as extensive as it could be, and it doesn't get diagnosed. So the other thing is it can often be transmitted by the respiratory route. People can have respiratory symptoms when they first get sick. They can have respiratory symptoms for 4 days before they ever show a rash. So they can be transmitting the virus before they realize that they have monkeypox. So this is just one example. So we have 36 deaths now from monkeypox as of October 2022. And this is just one example in Africa of people who died from monkeypox. One person here committed suicide because of the pain or because of the stigma, or we don't know exactly. I don't know why there. And then it's going to be high fatality rates for neonates, and including for pregnant women and babies and fetuses. Also, if someone's HIV infected and they have low T cell counts, they're much more likely to die from it. But it can kill perfectly healthy people. Here's a 27-year-old HIV negative, and he died after 9 days. So it's a serious virus. But it doesn't make such serious lesions in everybody all the time. Here's some pictures from the CDC website from the UK Health Security Agency, and you can see some of the lesions can be pretty small, but they can be severe in other people. So I think, as you can see, there's a number of lesions here. I think this would be recognized as monkeypox, but if somebody just has a single lesion or two, I'm not sure it's going to be recognized. Dermatologists may see this, gynecologists may see it and not recognize it for monkeypox. And challenges for diagnosis, these poxviruses, the one that's most frequently confused with in Africa is chickenpox, which is a herpes virus. And it can also be confused with other herpes viruses, herpes simplex 1 and 2, especially for the genital lesions. And then it can also look like secondary syphilis. So here's monkeypox on the right and secondary syphilis on the left. So there can be some confusion for identifying it. So, okay, what about vaccinations? And of course, this was mostly focused on smallpox, which is variola virus. So the first, hundreds of years ago, they used the smallpox scabs and used that to vaccinate people. That was called variolation. They would use the virulent pathogen for vaccine, which, had a higher fatality rate. And then Edward Jenner came up with the idea of using this pox viruses they got from cows. And that in North America was passed through cows and horses for hundreds of years because we couldn't do tissue culture, right? So we could only grow viruses in animals. So this virus that's been handed down over centuries is called vaccinia virus now, and cowpox is a distinct virus, nomenclature, right? Taxonomy. So, and we have had, anti-vaxxers. This is from the 1802. People were frightened to get the cowpox vaccine because they would turn into cows and have all these awful things happen. So anti-vaxxers have been around for a long time. So the vaccination for smallpox is a live vaccinia virus, closely related virus. And vaccination was stopped in the US in around early 1970s because of the severe side effects in some people. So most people that are under age 50 in the US now are not vaccinated, but military personnel are vaccinated, first responders, everybody in my lab is vaccinated. And we've also done studies that you can show vaccination right before or just after affords some protection. So this was a good news for a naive population if there was a bioterrorism or biowarfare event where we need to vaccinate people post-exposure. So this is the smallpox vaccine. It was dried calf lymph until 2007. That was the vaccine that was used. And then ACAM, they came out with ACAM 2000 in 2007. And so that's a tissue culture-based vaccine that we use now. And this is what the lesion should look like. You have a live virus growing in your arm for, they say day 21, but I mean, last time I've had about 5 of these, and I had live virus in my arm 6 weeks after the vaccination. So this is what the vaccine should look like and how it should develop. And you end up with a small scar, usually in your upper left arm. And it's great. And this is the vaccine that was used to eradicate smallpox. So the most effective vaccine success story ever. We eradicated the disease, but it's also the most dangerous vaccine that we've ever had. It can spread in the human. It can cause a generalized rash. It's really bad. And anybody who has eczema or some inflammatory skin condition should not get this vaccine because it can spread. Encephalitis, pericarditis, progressive vaccinia. If the person's immunocompromised, the virus can grow unchecked and it can kill the fetus. So here's a picture of some generalized vaccinia. It is rare, but way too common to be a general vaccine for the general public. And this is accidental infection in the eye, auto-inoculation. So, people aren't that careful. You can touch your lesion, scratch your arm and not even notice it, and then touch your eye. And then these people had eczema vaccinatum. So because of the eczema condition, the virus can grow very well in their skin. And neither of these individuals was vaccinated. They caught it from someone in their household who had been vaccinated. And then progressive vaccinia can occur in people that are immunodeficient, and the virus can grow unchecked and kill the person. So fortunately, there are some drug and antibody treatments available that are being used now, and these will work for all the viruses in orthopox virus. So vaccinia, cowpox, monkeypox, and smallpox complications. We assume they'll work for smallpox. They haven't been tested in smallpox not thoroughly. We've done most of the studies in monkeypox under the animal rule. So cidofovir works, and that can be used intravenously. Brincidofovir is an oral available one, but it's easy to grow out resistant pox viruses. Unfortunately, we can show that in tissue culture. And then there's ST246. That's a new drug in TPOX that was approved by the FDA in 2018. And we have some antibodies available, hyperimmune anti-vaccinia serum that's available. So we need to make better poxvirus vaccine options, right? So the main one that's been done is the use of modified vaccinia ankara. So this is a vaccinia virus strain that has 6 large genome deletions, so it can't replicate well in mammalian cells anymore, so it's much safer. But it has 6 large genome deletions, so all those antigens are missing. So this is being developed for cancer and for other vaccines. It's approved for smallpox. This is the one that's recommended for monkeypox and is being used now. It can be given subcutaneously, and they're also splitting the doses to do intradermal inoculations because they can use a lower quantity and the supply has been limited. Other pox viruses that have been used, the fowl poxes, canary pox was used for HIV vaccines. So that was the only HIV vaccine success story. It wasn't that good, but that was the protection for HIV. And then I'll talk about targeted deletions in virulence genes as one of the possibilities for making a better pox virus vaccine. So modified vaccinia ankara is the Jynneos vaccine. And so, it's really safe because it doesn't grow in mammalian cells very well. But it's not as protective against monkeypox. And that was shown in 2013. It doesn't induce the same antibodies that the full vaccinia virus does because it's missing so many proteins. It induces apoptosis in dendritic cells and prevents good antigen presentation. And so Bavarian Nordic is the one that has made the Jynneos vaccine, and they have a 2-dose regimen because you have to give 2 doses to get a really good immune response. So we really do need improved vaccines still. So my work has been in understanding what makes these viruses so virulent and how do they take over the body and turn off the immune response for long enough so that they can replicate and spread. So I did a lot of bioinformatics work looking at the viral genomes, and we published the poxvirus orthologous clusters back in 2003. And at that time, we had about 22,000 genes. And so we looked at identifying which genes were completely conserved in all the poxvirus, and we found 49 gene families that were completely conserved, from entomopox that infect insects all the way up to all of the mammalian tropic poxviruses. So these we identified as essential genes. Then there were another 41 gene families that were conserved in chordopoxviruses, so required for replication in vertebrate hosts. And then there's a large number of less conserved genes that can be virulence factors and host-specific factors. And then I realized that there was also a subset of these that were highly conserved in mammalian tropic poxviruses, but not in all the chordates. So they weren't conserved in the viruses that infect alligators and fish, other vertebrates that don't have as highly developed immune system. So I became interested in these, that they might be involved in regulating the specific immune response in mammals. So one of those genes that I began looking at was A35, Vaccinia A35. And it's highly conserved in all of the mammalian tropic poxviruses. It's novel. It's not found in anything else. Still, there's like almost no sequence homology to anything else in the world. I think a bacterial transposase was the closest thing I could find. Completely unknown function. And it's a very strange hydrophobic protein. So I began looking at this gene to try and understand what it might be doing in the virus life cycle. So the first thing, here's the life cycle. So first thing you do when you want to know what a gene does is you knock it out, right? So here we replaced the A35 gene with GPT, and here you can see plaque formation. So here's the wild type normal virus, and you can see it makes these holes in the monolayer, these white holes, the dark monolayer is stained here. So a plaque is the hole in the monolayer where one virus spread and infected the cells and killed them. So here, this is a normal plaque phenotype for the wild type. You can see how big they are. The A35 knockout is also capable of making these large plaques. So, and we looked very carefully, it replicates normally, it spreads normally in tissue culture, and it makes all the normal forms of virus. So I actually quantified the different types of the intracellular mature virus, the extracellular envelope virus. So it didn't seem to affect replication. And we looked at replication in all these different cell types and all these different animals to see if it might be affecting replication, including in mouse, nose, lung, blood, and brain 1 to 3 days post-infection. So before the specific immune response has time to kick in. And we didn't see any difference in replication for A35. And this is good. It's better for vaccination where you actually need to grow the virus. So it's not, replication defective, which makes it difficult to grow. So you could grow it up for vaccination. So the next step was to look and see if there was a function in vitro, since we couldn't find any phenotype in tissue culture in vitro. So we wanted to look in vivo, especially, as I said, I was interested if it was regulating the immune response. And it's a very nice challenge model that we have in poxviruses. You can just do intranasal installation of the virus and then watch body weight and mortality as readouts. So here you can see we have the mouse weight on the y-axis here in days post-infection. And these mice were challenged with saline, with the A35 deletion mutant, or with the wild type WR here. And you can see that with the wild type virus, these mice die. They lose weight and die. And but the A35 deletion mutant here, almost the same as PBS. And that was with 1,000 PFU per mouse. So deleting one gene, the A35 gene, made this huge difference in the difference in virulence and between life and death in these mice. And so we did a titration with different amounts of virus. Here you can see this is the 10 to the third I just showed you. Go up to 10 to the fourth, you get a little bit of virulence from the A35 knockout. 10 to the fifth, here again, you've got an intermediate amount of virulence. We had to go all the way up to 10 to the sixth to get it, the A35 knockout, to be as virulent as the wild type. So that's about a 1,000-fold increase, or 1,000-fold increase in virulence because of the A35 gene, and it doesn't affect replication. The other mutants that have large changes in virulence is really because the virus can't replicate. But this virus can replicate fine, but there's a huge change in the virulence in animals. So I was interested in looking at the immune response. So you can see here, I used 500 PFU, so half the dose here, to look at the immune response, because I wanted to look at the immune response, not at a difference between really sick animals and healthy animals. So I went down and used less virus for these infections, looking at the immune response in animals. So the hypothesis was that it's A35 is suppressing the immune response. And so I wanted to switch to see if I could find some in vitro immune assays where I could see a phenotype of A35 to start to pick apart what A35 is actually doing. And here you can see we have an in vitro model of antigen presentation where we can isolate antigen-presenting cells and infect those either with the wild type virus or the A35 knockout, and then pulse them, in this case, with guinea pig myelin basic protein as the antigen, and watch their ability to present antigen to this cognate T cell line. And this is MHC class 2 presentation. And if this antigen presentation occurs in this model, the T cells will make interleukin-2, and the T cells will be activated and then activate the antigen presenting cells to then make nitric oxide. So we have two readouts here where we can look at the effect of A35 on the ability of this antigen presentation to occur. And what we found here on the right, we've got IL-2 with increasing number of antigen presenting cells. You get increasing amounts of interleukin-2 up to a point where you reach a plateau. And so here you can see the uninfected cells present antigen very well and induce a lot of IL-2. And then the A35 knockout virus also can reduce the amount of IL-2 made, but if A35 is present, you get a statistically significant reduction in the amount of IL-2. So there's some other gene that's also interfering with antigen presentation, but the A35 gene is also very important in reducing IL-2 production. Not totally responsible for it, but important for it. And here you can see similar with nitric oxide production. So the viral infection decreases the nitric oxide production, but A35 is also makes a statistically significant difference in the amount of nitric oxide that is produced. So from this, we knew that A35 was able to inhibit this antigen presentation and these responses. So we looked at a large variety of other cytokines and saw a very similar pattern here, where the uninfected induces the most, the A35 knockout is decreased somewhat, and then with A35 present in the wild type virus, we get an even greater decrease. So you can see this pattern across multiple cytokines. Not IL-18 though, right? Because IL-18 is there and ready to go, so it can't really inhibit it. Interestingly, poxviruses encode a specific protein to block IL-18. So because A35 can't interfere with that, the poxviruses evolved a different strategy to take care of IL-18. So A35 is decreasing this immune responses and all these cytokines that are produced in response to this antigen presentation. And so we used the antigen presenting cell to present to the T cell, and then we had a bioassay for CTLLs. So we checked carefully to see if A35 was affecting these two T cell types. I won't go through all those studies, but we couldn't find any effect of A35 on these. So we began to focus on what A35 was doing to the antigen presenting cell specifically. And so we wanted to know if it was a difference in infectivity, if it was killing these antigen presenting cells, what it might be doing. So we looked carefully at these antigen presenting cells and to see if A35 was affecting the infectivity. Here you can see the uninfected cells, and here are the A35 and wild type infected cells expressing viral antigens. And you can see there's no difference between the A35 knockout and the wild type in the amount of viral antigens being expressed by these cells. There was no difference in apoptosis, there was no difference in metabolism, there was no difference in replication, and there was no difference in the response of these cells to interferon and LPS stimulation. So it didn't affect stimulation through LPS or interferon, but it did affect it through the T cell receptor. So, so we began to look specifically at what was going on with these proteins involved in antigen presentation. We looked at class 2 and also the antigen, and some reports are that class 2 gets decreased a little bit, but we really couldn't find it very much. So we didn't, like, what does this protein do? So we started looking at this antigen in the cleft of MHC class 2. And so this is inside the cell. Here's the endoplasmic reticulum and how MHC class 2 transits through vesicles to the surface. And this CLIP peptide, the CLIP invariant chain is degraded, and it leaves CLIP peptide in the cleft. And then antigen, DM, is a protein that catalyzes the removal of the CLIP peptide and loads antigen into the cleft, and then transits to the outside surface of the cell. And what we found out doing flow cytometry, looking at antigen in the cleft on the surface, is that A35 decreases the amount of peptide in the cleft on the surface of these cells, and it increases the presence of CLIP on the surface of the cells where it really shouldn't be. So we found out that A35 is inhibiting this antigen presentation through blocking this addition of the viral or whatever peptides, antigenic peptides in the cleft of MHC class II. So we don't know exactly how that's occurring. And we wanted to look and see if A35 was present in these endosomes where this would be happening. So we did immunofluorescence to look for A35 in the endosomes. And here you can see two cells. You can see the dark nuclei here. And this is Rab7, which is an endosomal marker for late endosomes. Green here shows the A35, and here's the merge. So you can see that A35 is present in these late endosomes. It's also present in intermediate, least present in the early endosomes. So A35 is in the right location to be inhibiting this antigen presentation in these vesicles also. So we wanted to know, exactly which stage A35 was involved in. And so we've looked at A35 effects on trafficking and acidification of the vesicles, on proteolysis, and we haven't been able to find anything. We've looked at a number of different things. It doesn't seem to affect the trafficking or acidification or proteolysis. And what we have seen is just a tremendous change in the composition of proteins in the endosomes. So, and that's between wild type infected and A35 knockout cells. So A35 is somehow majorly affecting the protein composition in these endosomes, but we don't exactly know. We've tried co-immunoprecipitation, we've tried a bunch of different things to see if we can identify what it's interacting with specifically, and we have not been able to do that. But we know it's got a major effect on proteins in the endosomes. So now I'm going to talk about zooming back out to look at the animal models. So we predicted, or the data suggests, that A35 is blocking this antigen presentation to the CD4 helper T cells. And these CD4 helper T cells help B cells make antibodies, and they help stimulate the CD8 positive cytotoxic T lymphocytes. So I predicted that A35 would decrease both antibodies and T cell responses in these mice. So we went back into the mice and began looking at this, and we found out that indeed A35 causes a significant reduction in antibody production, vaccinia-specific antibody production, and this is B5R, one particular protein that we looked at. So A35 does decrease antibody production in infected mice compared to wild-type virus, compared to the knockout. And then A35 also had a very significant decrease in the number of virus-specific interferon gamma secreting T cells, a tremendous difference in the number of T cells that were responding to the virus. So A35 does block the immune responses in vivo in these mice and can make the difference between life or death. So, so the poxvirus vaccines, we know that A35 blocks the immune responses. We know that removing A35 makes a much safer virus. And we wanted to see if A35 deletion mutant could actually act as a vaccine. So we did that study. We immunized mice with either the wild type virus or the A35 delete. So, small enough dose that they would have an immune response, but not be sick. And then we challenged these with virulent wild type virus, the WR, which is a neurovirulent strain. And I think we started off at 400 LD50, 500, so all the way up to 750 LD50 we've looked so far. And in each case, the A35 knockout is as good as the wild type virus for protection from virulent challenge. You can see this is a strong challenge that all the mice die by day 4. I'm showing you 450 LD50 and 600 here. We've been up to 750 now. So the A35 knockout makes a very good vaccine and is highly protective, at least against the WR virus. We haven't challenged it against monkeypox. I would like to do that. So the A35 we've shown is a virulence factor. And the A35 deletion mutant virus is much safer, about 1,000-fold in mice, really a tremendous difference. And we know that A35 blocks immune responses, both in vitro and in the mice, B cells and T cell responses. The A35 deletion virus is more immunogenic. You get a better immune response. And we did this in both the wild type and in MVA, the Jynneos vaccine that doesn't replicate, that is much safer. And we even get better class switching. We published that in Vaccine a few years ago. I've also done this with a cancer vaccine that I was working on for mesothelin for pancreatic cancer. And we did show we got a better immune response to the cancer antigen. Unfortunately, we couldn't protect the mice with this vaccine, but we didn't use a checkpoint inhibitor. And so I would like to go back and do that with a checkpoint inhibitor and see if we can get much better protection of mice using the A35 deletion mutant. So the A35 deletion mutant is highly effective as a vaccine, all the way up to 750 LD50 with a virulent poxvirus. So, and the other really good thing is that A35 deletion mutant has all the same antigens as the original vaccinivirus vaccine, whereas the MVA is missing 6 large, genome deletions of what's available to induce an immune response to. So, the only thing it's missing is A35 because it's the A35 knockout, and that doesn't appear to be much of an antigen anyways. We haven't seen it in immune responses. So I would like to thank a number of the people in my lab who have worked on this project over the many years, and funding that I've had, especially from NIH and Beringer North Carolina Biotech Center. So I want to acknowledge that. So I'm happy to take any questions now. Thank you.
Thank you so much, Rachel. Very excellent lecture, and both very comprehensive and at the same time a lot of insights on some molecular basis, and how we can improve vaccine and decipher virulence. And so this presentation is open for questions. So Jesse, also we have, oops, I lost my
There was a question from Mark. I saw something pop up.
Yeah, you have question. You have some questions.
Okay. Have you seen the most vulnerable populations to be the same worldwide? So monkeypox used to jump into humans from animals as a zoonotic infection, and it didn't spread very much human to human. And what we're seeing now, and probably since 2017, is sustained human-to-human transmission. And it may just be because of it's spreading, especially with men who have sex with men. So it may just be that it got into that population and it can spread better by that route. But the other possibility is that there are actual genome changes that are making it more transmissible human to human. So we can't really distinguish those two. And there's a huge number of mutations that have occurred as it's adapted to human. And many of them may be due to the ApoBec3 virulent gene in humans, the effect of that enzyme on the virus genome. And of course, older people are more sensitive, younger people, babies, children are more sensitive. So it's the opposite of COVID. And anybody who's in contact with animals or men who have sex with men, especially with a large number of partners, are going to be more vulnerable. The next question I see is, do you think A35 may bind directly to HLA-DO or HLA-DM? Oh my gosh, we have tried so hard to demonstrate an interaction between those two, and we have not been able to. Some of the antibodies that are available for those are not very good, and we could do some genetic alterations and look for interactions between those two, but haven't been able to do that. That's what we were thinking, and we thought we would be able to demonstrate that with co-immunoprecipitations, but we haven't been able to do that. And again, the antibodies are not great, what's available to detect these proteins. And they're very low quantity too. And A35 is a low quantity protein. It's made early and shut off during the virus life cycle, but it lasts, for more than 24 hours. We looked at its stability, so it's fairly stable, but it's not very high quantity. It's also a very hydrophobic protein, so it's really difficult to work with. The other problem is that it's one of the substrates for the poxvirus cytoplasmic redox system. So it's got disulfide bonds made in the cytoplasm, and that hasn't been reconstituted yet. So it makes it difficult. We can't just transfect A35 into something else and expect it to work, because it won't be disulfide bridged like it would be during a poxvirus infection.
Thank you. So you see that you have questions now on the. I do not see them very well. So I mean.
I saw 2 questions in the webinar chat, and I have answered those.
No, but you have on the Q&A. You have questions on the other.
Okay, I see. Okay, Mark Kaplan. Does A35 induce HEC point molecules like PD-1, et cetera? I don't know that one. Yeah, I don't know that. And then did I get the fact A35 interferes with immune presentation and still is a good vaccine? Yes. So that's been, people have wondered about that for years. Like, so we know that when you get infected with a pox virus, they say you have lifelong immunity afterwards, right? So smallpox, monkeypox induces lifelong immunity. So it does induce good immunity, but you're right. The virus is still blocking the immune response while it's infecting you. And what we found with A35 is it really delays the immune response. So eventually it catches up a month later, but you know, the virus doesn't have to turn off your immune response completely. It only needs to turn it off long enough to be able to replicate and spread, right? So that's what viruses have to do to exist in nature, is replicate and spread. And so it doesn't, it doesn't have to stay in you forever as a persistent infection, but it has to stay long enough. And A35 decreases and delays the immune response. I'm getting a pop-up from my university about software. Okay. So my work has been done in a biosafety level 2 lab. I did get approved for BSL-3. We have BSL-3 for COVID where I am, but the BSL-3 select agent monkeypox, I got approved for work in Duke, but we don't do that at East Carolina University in my university. I see one that says antigen presentation, but is there a question on that one? Okay. You mentioned that you can select for cidofovir-resistant virus in culture. Yes, quite easily. Are the cidofovir mutation in the polymerase region? I don't remember, but I think so. And they were quite easy to make. And the good news about the cidofovir-resistant mutants is that they seem to be attenuated. I don't remember exactly how they were attenuated, but yeah, and I think they're growing mutants out for the ST246, the TPOX also. So we really do need to keep working on drugs. And and I'm concerned that they're not recommending treatment for long enough in these monkeypox infected patients. They're treating them for a short period of time, but short for relative to the infection time. And so, I mean, the best way to grow out resistant mutants is to treat them with a drug, not strong enough and not long enough. And so I'm concerned that we are currently encouraging the development of drug-resistant monkeypox viruses now. Alan Schmaljohn, hi. So A35 notwithstanding, vaccinia is highly immunogenic, both antibody and T cells, yes. But this is typically a heterogeneous viral population. That's true. Perhaps containing A35 deletion mutants. ACAM 2000 is clonal, no change in A35. Okay, so the original virus was vaccinia virus Dryvax. And in 2007, they released a tissue culture-based ACAM 2000, which is what is used for vaccination now in lab workers. They're not using it much at all in the monkeypox outbreak because they're too concerned about the virulence. But ACAM 2000 does have A35 in it, and it's pretty similar to the Dryvax. But Dryvax was an incredible heterogeneous. We did sequencing in Dryvax, and like every single virus in that virus preparation for the vaccine was different. Every single one had mutations. It was just incredibly heterogeneous. So the ACAM 2000 is clonal and looks very much like the Dryvax original population and has similar side effects. So it's still pretty high virulence. So the ACAM 2000 has a lot of residual virulence, which is why they're using the Jynneos vaccine, the MVA-derived one, but that has the 6 large genome deletions and doesn't induce the same immune response as the original vaccinia virus.
Thank you so much, Rachel. I believe that you can go back to the chat. Did you answer the one from, did you see the question from David Ostrov? Oh, yeah.
So are the dominant class 2 restricted T-cell epitopes known from monkeypox? I know that work has been done on the dominant T-cell epitopes in mice for a number of different viruses. And they're different for, if you look at BOB-C and C57 Black 6, they're different epitopes, right? So I don't know specifically about monkeypox. And because humans are outbred, I think you're going to have quite a variety of them. What works for one human isn't going to be the same what works for another human, because not only are we outbred, but all of the rearrangements for the T-cell receptors are mutation driven, combinatorial between variable diversity region and then non-template derived nucleotides being added and also chewed off the ends. So I think there's going to be a wide variety of epitopes that work for different humans. Are drug treatments readily available around the world? No. There's a real problem with the drug supply and vaccines being available, especially in lower income countries. And even in the US, the brin-cidofovir is hard to get. I think cidofovir is more readily available, but that has to be used IV. So there really are problems with drug supply and vaccine supply worldwide. What do I see as the next steps in research to control monkeypox? So luckily, fortunately, the cases are going down, at least in some of the developed countries. We've got education warning people to be careful because we know it's circulating, especially with men who have sex with men. We need to increase, surveillance. I think monkeypox has clearly been spreading in the world for years without people recognizing it properly. I think there's a lot of more mild cases that are getting through, and we see the more severe cases. And so we need more drugs, better drugs, especially because we can easily make resistant mutants. I think we need better vaccines. So I should disclose, I have a patent on the A35 knockout virus, but no company has picked it up because the Jynneos was already used, and it costs so much to get a vaccine through to approval, hundreds of millions of dollars, that nobody has used the A35 knockout virus yet. I would really like to see it used. I don't care anything about making money on it. I just think it's a great vaccine and would like somebody to use it. I am working with some people doing some cancer vaccine studies who are using the A35 knockout. So hopefully, because it's a better virus, it grows well, and has much slower virulence because it doesn't block the immune response. So yeah, we need to have good surveillance worldwide. Wherever viruses are jumping into humans, it threatens the whole world, right? Because we have so much travel now. So rich countries can't ignore what's going on in lower income countries because they directly affect our health. So we need to be watching worldwide what's going on, and we need to be providing drugs and vaccines to countries where these viruses are jumping to protect the whole world, not just certain countries.
Finally, Rachel, you can go back to the Q&A.
I see one here. My own work suggests a bivalent, perhaps RNA vaccine containing L1 and A33 might be highly effective. Hooper did quadrivalent. Yeah, there's a couple of vaccine that look pretty good using 2 or 4 of the poxvirus gene as antigens, and they seem to provide pretty good protection. Certainly you would only have an immune response to those 4 antigens, so it wouldn't be as good as an A35 knockout. or as good as the Jynneos, but it might be enough to provide good protection, and you don't have to worry then about having a live virus vaccine. Of course, the RNA vaccines are very, unstable, right? You have to keep them at -80, and so that would be a problem for bringing that into areas of the world where there isn't good refrigeration available. Yeah, and that's with the COVID vaccine. The only gene that we've used to target is spike, and that's only 13% of the SARS-CoV-2 genome. So I think we really need to have several genes in the COVID vaccines, and I think that would help get better protection against different mutants.
Thank you. I believe that we will close these lectures. Many questions, actually, very interesting discussion. Well, thank you again, Rachel, for really a wonderful lecture. Thank you, everybody. Again, I mentioned that this is a very interesting partnership between Global Virus Network and USF, which really illustrates, in my view, and this talk has been perfect for this, how basic research, molecular-based studies can really inform global health strategies and how help control infectious disease. And to close, I forgot an important point, is that USF will become the first regional headquarters for the Global Virus Network, in charge of coordination of the activities for the whole Florida and Southeast US. And this lecture again and again has very well illustrated this, with attendees from USF and from the Global Virus Network. Thank you again, Rachel. Thank you all. Wish you a very good day. Bye-bye.
Thank you.
Thank you, Jessie Casanova.